Many of us in the audience know Professor David Cooper very well. David is a great mind person, a great heart, a great doctor, a great scientist, researcher, a great leader, a great inspirer, and one of our great friends. David, you and your wisdom are always in our heart, and your mission is now continuing by many of us. We always miss you, David. It's my great honor to give this memorial lecture for Professor David Cooper. My talk is the update on Chulalongkorn mRNA vaccine development at Chula Vaccine Research Center in Bangkok. In the last 2 years, we have been facing a serious pandemic of COVID-19. And all the global society, healthcare system, and economy has been hit very hard up until now and maybe a few years ahead. One of the record-breaking in science is the COVID-19 vaccine development. Never before in the history that against only one pathogen, scientist, researcher was in academia, and the biotech company has been working on COVID-19 candidate vaccine. More than 200 vaccine candidates. And now at least 113 vaccine had been on clinical trial or finished. Some of them has been authorized, 16 authorized, 9 has been approved for clinical use. So it's really a record-breaking in terms of the speed of the vaccine development, the number of vaccine development, technology platforms, particularly new technology platform. 3 of them has been authorized or approved. Among them are mRNA vaccine, viral vector vaccine, and DNA vaccine. And the efficacy is also very remarkable, particularly by the mRNA vaccine. And now, if you look at the vaccination tracker globally, up to 10 billion doses of vaccine has been delivered globally, with the average of 35 million doses a day. A half covered by a traditional technology, which is also saved many people life, which is inactivated vaccine. Another 2 billion doses covered by mRNA vaccine, another 2 billion by viral vector vaccine. But the challenge remains ahead in terms of inequity to access COVID-19 vaccine. And you look at the dark green, the darker green is mean the area that has been the higher proportion of population has been full vaccinated. But if you look at the big hole in this continent, in the African continent, less than 20% of people had been fully vaccinated. In some countries, even less than 5%. So the evidence is so strong that no one is safe until everyone is safe. So now we are facing Omicron, a very highly transmissible variant, and many more to come until we make everyone safe. Then one will be safe. Chula Vaccine Research Center, we have been working on another technology platform, subunit protein, DNA, viral live particle. Right now, we move to mRNA vaccine technology platform, and with the goal to make it more accessible in low and middle income countries. This is because of in the past several years, we have been collaborating with one of our good friends, Professor Drew Weissman. He's one of the pioneers of mRNA technology. And I'd like to invite many of you or all of you that are interested to listen to these great three scientists who discover this technology platform, mRNA and lipid nanoparticle, who are our honorable speaker. Recently, the three of them received Prince Mahidol Award 2021, and going to give us in person a talk on this technology, how this technology or this discovery can help global health fighting against pandemics. Now, I'd like to share with you here is the milestone and the timeline of our Chulalongkorn mRNA vaccine had been developed so far. So in the early last year, even before we have learned that mRNA vaccine like Pfizer and Moderna work the best in terms of efficacy, up to 95% efficacy against COVID-19 symptomatics. And we start working on that, and it has shown a very strong induction in animals, small animal, and in non-human primate. And then at that time, we don't have manufacturing technology yet in Thailand or even in Asia. So what we did is we worked with two CMO in the US and to making our clinical lot. And it's been taken quite long because we don't have that technology. So we spend overall by negotiation, finding the right CMO and starting making it. We start phase 1 in mid of June, and then we now had finished phase 2, and the Thai biotech company BioNet Asia already got all the technology transfer and start making locally clinical batch for phase 2-3 trial in the very near future. The difference between our immunogen design versus the authorized Pfizer and Moderna vaccine, the same technology platform is a modified nucleoside techniques. The antigen is different because most people believe that you need to stabilize pre-fusion spike protein to make it as stable. But in our case, is a non-stabilized spike mRNA protein, and the lipid nanoparticle also from different formulation. So Chulalongkorn is right now among many other mRNA vaccine, both against COVID and non-COVID in entering phase 2. Now I'm going to share with you the clinical results. As you can see that we have finished complete the enrollment and then finished the interim analysis of 2 phase 1 study and 1 phase 2 study. So we very grateful to all of the dedicated volunteer and our development team. This is to summarize in brief that how the phase 1 study design has been performed. So we have seen the phase 1 report of Pfizer Moderna very clearly that the vaccine is very immunogenic. So we start with only small sample size to identify those that in between lower than Pfizer, in between Pfizer and Moderna, and to identify optimum dose for our immunogen. So we start with the 10 and then move up to 25 as a dose escalating design into 50 microgram. This is a 2-dose schedule, 3 weeks apart. The primary endpoint is safety, and the secondary endpoint immunogenicity. And we also have performed an exploratory comparative study with the Pfizer vaccinated cohort and also with convalescent sera. I'm sorry that this is a quite busy slide, but to illustrate that vaccine and in phase 1, it's in general it's safe, no serious adverse event. It's a dose dependent, and also the second dose will have more both local and systemic AE. Most of the AE are in green, so it's mean mild. A few in moderate, and very rare in severe grade 3, but all are recover within in average of 2.5 days. And you can see that the oldest group has a lower. This compare 10 microgram younger and oldest group, and 25 microgram in all AE. That oldest group has lower, significantly lower AE. And now look at the immunogenicity results, and this is the anti-spike. It's a white type spike without dipolin. So you see that this is a dose dependent, 10 microgram in blue, 25 microgram in orange, and 50 microgram in pink. The left panel is the youngest group, 18 to 55, and the right panel is older age group. And you can see that the lower dose peak later. So at the day 50 is 4 weeks after the second dose, compared to the higher dose of 50 microgram, reaching the peak faster. In terms of surrogate neutralization assay, this is the RBD ACE2 binding inhibition assay. And you can see that low dose also be able to reach at day 50 above 90%, but it at day 25 is not at that point yet. 50 microgram have reached more than 90% seen earlier. And this is compared with in green is Pfizer, 93%, and the convalescent sera is 76%. This is the result of the live virus microneutralizing assay. Look at the 4-fold rising, or what we call the seroconversion rate. If we look at the 1 week or day 29, 1 week after the second dose, only 50 microgram of Chulalongkorn and BioNTech Pfizer vaccine reach 100%. But after day 50, all doses reach 100% seroconversion rate. This is the comparison between dose of live virus neutralization test. The units is IU per mil, compared with the two age group, adult and elderly. It's a very similar pattern of the kinetics. The lower dose peak a bit later, and the lower dose have a significant drop when you vaccinate in the older population. 10 microgram has 5-fold drop in elder people, 25 microgram and 50 microgram drop about 2.5-fold. And this is the neutralization, the GMT of Pfizer, 500, and the seroconverter convalescent sera is 260. And this confirmed by another assay, a functional assay, pseudovirus neutralization test, similar finding, and the big drop getting older in the lower dose, and lesser drop in the mid dose, and further lesser drop in the higher dose. And as compared to the green is a Pfizer BioNTech vaccine, and the purple is the seroconverter. And as we have seen in the animal study that the magnitude of antibody titer neutralizing again white tide is so high. Even it's dropped significantly again valence, but it's still high enough to cross neutralize alpha, beta, and delta. In particular, in the higher dose of 50 microgram, lower doses didn't perform very well against delta. And this confirmed again with, that's the live virus neutralization. This is the pseudovirus neutralization assay. I highlight the one that outstanding is in the adult beta is very high against beta, which is one of the most difficult to neutralize using white tide based vaccine. And the delta also stand out very significantly in terms of 50 microgram dose as compared to Pfizer and Moderna, Pfizer-BioNTech vaccine in green. How about the T cell response? In this case, gamma interferon ELISpot assay result showing as the spot forming gamma interferon producing cell or spot per million PBMC. On the left hand panel is the younger age group, and the right-hand panel is the older age group. It's very similar to what we have seen in antibody response. In the older age group, the lower dose have a twofold drops in the 10 microgram and 2.74 drop in 25 microgram, but very consistent in a 50 microgram. And all of these profile CD4 and CD8 specific response is showing at the Th1 predominant with the Th1-Th2 ratio greater than 1.8 or 2 in all doses. So 50 microgram by our DSMB recommendation had been selected to go for phase 2 trial. Now, in terms of the phase 2 trial, is a RCT, evaluator blinded, 4:1 ratio. So this is the age group is 18 to 59. 120 received Chulalongkorn 50 microgram 2 doses, 3 weeks apart intramuscularly. Another 30 received placebo. It's a normal saline for 29 days, average of 1 month, and then all switch to receive Pfizer-BioNTech vaccine 2 dose, 3-week interval. Safety is a primary endpoint, and the immunogenicity is secondary endpoint. Here are the adverse events. You can see that first dose, there's no fever at all. Both the placebo and the control. Interestingly, the placebo effect to induce headache related is higher in the first dose in placebo arm, almost double, 37% compared to the Chulalongkorn first dose, and 30% fatigue by placebo and 40% by Chulalongkorn. Fever at the second dose is high up at Chulalongkorn up to 24% versus 0 in placebo. Now, anti-RBD antibody response in blue is the Chulalongkorn placebo here in the middle, and the placebo after 29 days switched to Pfizer-BioNTech. So this is the result, and you can see that at day 29, day 20, and day 50, so 4 weeks, it's clearly that the geometric mean titer ratio against Pfizer-BioNTech is 1.84 higher with the p-value below 0.001. And this is the statistic analysis of at day 50. You can see that it's highly significant, about 1.84 of Chulalongkorn anti-RBD antibody against Pfizer-BioNTech. In terms of live virus neutralization, it's very clear again that it's higher, about 2.7 ratio against Pfizer, and p-value below 0.001. So this is the Chula-19 placebo, Pfizer, and zero converter. This is convalescent sera. And you can see that this is the p-value on day 29 of live virus microneutralizing test, 850 titer versus, this is not the IU per mil, this is the unconverted, but you can see the ratio is still the same, 2, 4, and 2.74 with the p-value on day 50, again, white ties below 0.001. It also confirmed again by the pseudovirus neutralization test. This is converted into IU per mil, again, white tie. It also, and also when we compare again Delta, this is compare Delta by pseudovirus, it's also 1.74 higher than Pfizer vaccinated sera. And this is the statistical result. You see that white tie, day 29, day 50, and then on day 50, 2.84 higher with the p-value below 0.001. And with Delta also, the ratio between Chula-19 versus Pfizer-BioNTech is 1.7. The p-value is 0.1. T-cell confirm what we have seen in phase 1, that on day 29, with more than 1,900 spot forming cell per million cell versus 800. And this is the ratio is 2.24 with the p-value below 0.001. Again, this is the statistical analysis comparison. So in summary of our interim results of both phase 1 and phase 2, the vaccine Chula-19 at 50 microgram, 2 doses, 3 weeks apart, intramuscularly, is well tolerated. There was no serious adverse event. Again, in terms of induction B-cell, specific B-cell response, Chula-19 able to induce high neutralizing antibody against white tie. And although, again, valent is a significant drop, several fold, but it's still high enough to cross-neutralize alpha, beta, gamma, and delta, in particularly delta. And also induce high T-cell response. Phase 2, Chula-19 is superior than Pfizer-BioNTech vaccine in all specific immunogenicity tested. Result had been shown. How about the next generation vaccine? Again, new variants. We have now facing one of the most transmissible variants, Omicron, and also escaping significantly again the current vaccine, particularly 2-dose vaccine. So among 5 vaccine of variants, divided by the WHO, we have in green, it means we have prepared the prototypes immunogen of alpha, beta, and delta. And now we are working on Omicrons. Hopefully in the next few weeks, we should be able to test in animal. So this has been tested in animal. Based on the phase 1 results, the government has, the cabinet has approved 2.3 billion Thai baht to support our Chula-19 mRNA vaccine development for the larger scale trial and for the legislation. And we have been working with our strategic biotech company in Thailand, BioNet Asia, has been listed right now among one of the 10 manufacturer company besides Pfizer and Moderna. So now we are working, the clinical batch made in Thailand by BioNet Asia has been finished and has passed all the quality tests. Now we're working with our regulator, the Thai FDA, to plan for the next step of clinical development, the late stage. Thank you very much for your kind attention, and thank you all the volunteers, my collaborator, both within Chulalongkorn University, within Thailand, and our great friends, Professor Drew Weissman and his team, and all the clinical team, collaborators, and the funder, the Thai government, the National Vaccine Institute, and the National Research Council, and 1/3 of the funding for phase 1 and 2 also coming from corporate and public donation. Thank you very much.